Well, let's see. The fundamental function of the kidney should have already astounded you that it's as complicated and interesting as it is starting to appear to be. We're going to see two more ways today, or three, that it's really very interesting. And its engineering is magnificent. Hopefully the video recording that you've listened to, most of you, is really helping you get a sense of how important this is and how valuable. We'll see a little bit as we go forward how the kidney acts like a homeostasis maintenance device that keeps our blood volume at the right level and keeps our blood osmolarity at the right level, both of which are crucial for life. That the osmolarity largely depends on the sodium chloride concentration. Those are the most concentrated and impermeable of the salts. And so we're going to talk primarily about sodium osmolarity today. And at the same time, preserving water for land animals is crucial. So let's start off with water. Question 1, can you all see it? It was supposed to open at 9 o'clock. Should be visible to you now. What can the kidney do to compensate for drinking too much water? Say you are in a water drinking contest. Now, there's going to be a homeostatic reflex if you do that, with its sensors and integrator and comparator and effector. The sensor and integrator, comparator, and the effector organs are called nodes in graph theory language. And the connections to them, like the neurons that connect one thing to another, we call those edges in graph theory, or arms in physiology. What are the different arms of the reflex, this negative feedback loop? And I use those terms to get you warmed up for the final project, so I hope that helps. Go. And homeostasis can involve the kidneys excreting salts and water in the urine. Some of you picked up on that, which would counterbalance the increased blood volume that drinking water would produce, because water is well absorbed in the stomach and just gets right into the body, into the bloodstream. So it increases our blood volume. And by excreting salts and water in the urine, we will return our blood volume back to normal, and our blood pressure, which might have been increased by the increased blood volume due to stretching of the vessels, that will also come back down. Now that's a side response to increased blood volume that we call the slow response. There's also a fast response in response to increased blood pressure, where we decrease our cardiac output by the baroreceptor mechanism. The main mechanism for returning excess water to nature is the aquaporin channel. The aquaporin channel, which brings us to question 2. We'll go into more detail here. Nascent urine gets concentrated at the end of the descending limb of the loop of Henle. Remember we talked about that for urea? And so does the interstitial fluid. Thinking of this picture as I read the question, where the darker blue interstitial fluid is 1200 milliosmolar solution, whereas up in the cortex, the osmolarity is near that of normal blood, around 300 milliosmolar. There's a boundary between the cortex and the medulla that is designated by an artery that crosses over between renal papillae called the arcuate artery. So below that, the concentration gets higher and higher as you get down towards the end of the papilla in the renal pyramid. 1200 milliosmolar. Small solutes are more concentrated deeper in the medulla, but then it gets dilute. The nascent urine gets dilute in the ascending limb of the loop of Henle. I'm thinking of this figure as I say that, which also shows the more concentrated interstitial fluid. How? How does it get more dilute as the nascent urine goes up the ascending limb of the loop of Henle? Go. Potassium chloride transporter of the loop of Henle is the main thing that we're talking about here. Sodium transport without water in the ascending limb of the loop of Henle leads to excessive solutes out in the interstitial fluid. Also, the urea that's coming out would increase the urea concentration, and that would increase the osmolarity gradient in the interstitial fluid between the nephrons in the pyramids of the kidney. In this diagram, we see too that there are actual sodium transporters that are active transporters and urea transporters that can add a final touch of increased concentration into the interstitium as the nascent urine gets down towards the end of the collecting duct where it empties into the calyces and the pelvis of the ureters. So the main point that I want you to get out of question 2 is sodium comes back out, water does not, so we end up with fluid that's 300 milliosmolar, even lower. It can be as low as 100 milliosmolar. As we get to the last question, we'll come back to this little thing right here, this sodium transporter or transport process that really allows us to kick sodium out at the end of what's called the distal convoluted tubule before it empties into what's called the collecting duct. Okay, but let's first go on to question 3. How are ADH and the interstitial osmolarity used to fine-tune urine osmolarity? And how does the peritubular capillary help preserve the osmolarity gradient? Before I forget, the opportunity to watch the video ends at class time from now till the end of the semester, starting today. So those who didn't watch it yet, I'm sorry if you forgot that, but that was the instructions and the way it was set up. And similarly, the one that was for last Friday we left that open on an exception basis because of being spring day. That was open till midnight last night. I mean, Saturday night. And so some of you might not have watched that in time to get credit for that. But don't forget to follow the directions to claim your credit, both for watching the video beforehand and only for that. And we will make note based on this question of who has attended and participated in digital dialogue. If you've entered any question, answer at all, you will get credit for today's session. If you've made any answer to this question, you will get credit for today's section. Okay, so antidiuretic hormone influences this process. Water comes out of the collecting ducts. If there's aquaporin in the membranes to let it out, because water doesn't get through cell membranes very well. It needs a protein channel. And so if there's a lot of aquaporin around, a lot of water can get out, and it will flow from this highly concentrated water nascent urine out into the low concentration of water, high concentration of salt, interstitial fluid. Let me say that again. The water is high in concentration and the salt low, coming into the collecting duct. And so water flows down its concentration gradient, i.e. by osmosis, water leaves the collecting duct and goes out into the interstitium because it's more salty out there. It draws the water out. The water flows down its concentration gradient, if you will. A better way to say it is activity gradient. And that's if there's a hormone around that stimulates the insertion of aquaporin channels so that water can get through the membrane. And it will really come out down in the bottom of the collecting duct, deep in the medulla, because the concentration is so high outside in the medulla. If there's not antidiuretic hormone, also known as vasopressin, in the blood, then we have fewer aquaporin channels, so the water can't get through the membrane, so it stays in the urine. So the urine keeps its high water, low salt concentration. 100 milliosmolar is low salt concentration. So we'll urinate dilute urine. The term that clinicians use is the specific gravity. The specific gravity is closer to 1 when it's dilute, because pure water is 1. But as you add more and more salt to water, its density increases, so its specific gravity increases. So the specific gravity of urine that has 1200 milliosmolar salt will be very high.
Question.
Yes.
From Joseph.
Go.
So from how I understand it, in order to block the passage of water, you have to remove the aquaporins from the membrane?
Yes.
And is that seems like it would take quite a while, like it'd be a slower response, wouldn't it?
Yes, because they'd have to be resorbed through an endocytosis process. It wouldn't happen in minutes. It would be a slow, steady process.
Okay. Cool, that was it.
Good question. And we'll bring that up a little bit more as we go on to the next question. Antidiuretic hormone, or vasopressin, released from, the D is missing, from posterior pituitary cells. That is the neural hypophysis, remember? Secretes ADH. This release is stimulated by 3 major stimuli, 2 from the cardiovascular system, there should be a comma after stimuli, and 1 in the hypothalamus. What are these 3 types of stimuli? And how does the kidney compensate for the derangement of homeostasis that are producing those stimuli? Go.
While they're doing that, I had one more question come in. It says, how does the medulla keep such a large osmolarity?
That's a great question, and let me come back to it in just a few more minutes when I talk about kidney punches. because the question is based on the point that this is just interstitial fluid. There's no barriers out here to diffusion. Why doesn't this salt just diffuse down its concentration gradient and equilibrate? It does when you die, just so you know, or when you have kidney failure. Well, you guys are really doing great today. I'm so proud of you. Okay. Ready? If the osmolarity gets too high, and by the way, that's plotted in this hard to see, this thumbnail picture of how plasma vasopressin goes up as plasma osmolarity goes up. If the osmolarity exceeds 280 milliosmolar, osmoreceptors in the hypothalamus stimulate interneurons to activate the hypothalamic neurons, which transport by fast axonal transport in vesicles through the infundibulum of the pituitary gland to the posterior pituitary release neurons, vesicles that contain vasopressin or antidiuretic hormone. And this hormone is secreted by exocytosis into the bloodstream and goes to the collecting duct epithelium, among other places. The other places, by the way, are indicated by this name, vasopressin. It means cause the blood pressure to go up by stimulating constriction of blood vessels. The arterioles, smooth muscle, constricts, causing the blood pressure to go up throughout the body. That's where it gets that name. But we're focused today on what it does at the collecting duct epithelium, which is its antidiuretic effect. Also, if atrial stretch receptors detect a low blood volume, they will stimulate the release of vasopressin. And that's going to help because we will keep more blood volume in response to, say, bleeding. Or if the blood pressure declines, the baroreceptors not only send a message to the cardiovascular control center to slow down the heart and speed up the heart and increase the blood pressure, but they also send neurons to the hypothalamus nearby, telling the hypothalamic neurons to release vasopressin. That will, as I said, have the impact of bringing blood pressure back up to normal, but also through the vasopressin effect, but also increasing the water reabsorption to conserve water through the antidiuretic effect of this same hormone. Okay, three different things. Now, this decreased blood pressure would also occur if you are bleeding. And it would produce a reduced pressure in the carotid bifurcation and the aortic arch where baroreceptors detect the blood pressure. That's a little different from the atrium, where a new type of receptor now detects stretch in the wall of the atrium. This is more of a volume receptor, we like to think. It is detecting the stretch, just like the stretch receptors in the carotid bifurcation in the arch of the aorta. But it's more of a, without increasing the pressure very much type of stretch. So it's more of a volume change than a pressure change. Anyway, these stretch receptors, those stretch receptors, they stimulate this, and these osmoreceptors stimulate this. There's two types of stretch receptors and an osmoreceptor. These stretch receptors we call baroreceptors because they detect the stretch due to pressure. These we call stretch receptors that decrease the stretch, that detect the stretch associated with blood volume. How does the kidney compensate for the derangement of homeostasis? Well, by increased water reabsorption. If you have too concentrated a blood, you need more water in your bloodstream. If you have too low a blood volume, you need more water in your bloodstream. So they stop urination, anti-diuretic hormone, by insertion of aquaporin into the membrane. Next question. Wait, before I go to the next question, I want to just reiterate something about aquaporin. It feels to me like I missed this. Yes, I totally missed this slide, and I meant to talk about it between these two questions. So let me spend one more minute on this. The vasopressin, or antidiuretic hormone, binds to receptors. It gets out of the bloodstream, stimulates the production of cyclic AMP, which stimulates the deposition of aquaporin into the apical membrane lining the lumen of the nephron. This would be in the collecting duct and distal convoluted tubule. Collecting duct and distal convoluted tubule. And that then lets the water through, and it can also get through the basolateral membrane through channels that are constitutively present. That means they're always there. And so now this membrane lets the water diffuse down its concentration gradient by osmosis, going up the solute concentration gradient, 300 milliosmolar, 600 milliosmolar. The water goes into the salty solution, gets into the blood.
What about 5 questions?
Can you summarize any questions? Let's see. Okay, let's take questions for a minute. We've got time.
Okay, so real quick, one of them is the kinetics of the aquaporin channel. Is it faster, slower to remove or place it in? Do we know anything about that?
It would normally be expected to be faster to place it in than to get it out.
Okay, so these two are pretty similar, but one says, how do those receptors on the previous slide, I think, differentiate between low volume and low pressure? Doesn't low volume cause low pressure?
Yes, low volume does cause low pressure, but it's a bit of a semantic point, because the atrial stretch will cause increased atrial pressure, but the atrial wall is so flexible, it's a little bit like a flabby balloon. And so they're not really detecting a pressure increase, think of Laplace's law, inside the atrium, as much as how much the atrial wall is getting stretched out. Whereas the carotid artery and aorta are very inflexible, and so pressure causes stretch without much change in volume. So they're thought of more as pressure detectors, whereas the atrial wall stretch receptors are thought of as volume receptors.
Okay, last one. Is aquaporin just a channel or does it make a channel? So I guess what is the definition of aquaporin?
Aquaporin is a channel which is formed by a protein that has a very narrow pathway. In fact, it's a magnificent study in molecular structural biology that water can get through a very narrow pore, but salt cannot. Water can get through very fast, but even protons cannot because of a gate in the middle of the channel that keeps charges from getting through. Only neutral water molecules can get through. So that's a magnificent molecule. And there's several subtypes of aquaporin.
That's it.
Okay, question 5. How does countercurrent flow in veins running alongside arteries in the appendages preserve the core body temperature? If you didn't already, go. I am muted by the host.
You're on mute, sorry.
Answer is, since the arteries bringing warm blood from the core of the body down to, say, the fingertips. Think of this as the fingertips or the end of your limb, the tip of your toe. Since they're bringing warm blood down and there's oftentimes cold air out, think you're outside in a snowstorm, you're in the Arctic, the heat from your body is oozing out through the tips of your appendages. It would be a lot worse if it weren't for the fact that arteries run right alongside blood vessels. So the arterial blood bringing warmth to your appendages can heat up the venous blood, bringing cold back from the tips of your toes. And that's called countercurrent. It preserves heat. This heat that might have got lost if they weren't running alongside of each other stays in the bloodstream. It's a principle that's used in chemical engineering. And the kidney uses it in a double fashion. Next question. In the kidney, the peritubular capillaries bring hyperosmotic plasma into the medulla, capturing the sodium pumped out of the ascending limb that we talked about previously. And then they absorb water, leaving the descending limb by osmosis to leave isosmotic, that should say urine, isosmotic urine, or to leave the urine isosmotic. Once again, what are the pumps in the ascending limb and why doesn't water follow? Go. The answer is these sodium chloride, potassium, NKCC pumps, secondary active transport pumps, are pumping sodium out of the ascending limb of the loop of Henle, as we said before. And that is resulting in concentration of the interstitial fluid because water doesn't follow, and diluting of the urine so that it gets clear down to 100 milliosmolar, even below the normal 300 milliosmolar of the plasma. The plasma coming in through the peritubular capillaries is picking up this sodium chloride that is being kicked out of the nascent urine in the ascending limb of the loop of Henle. So it's getting concentrated as it dives down in countercurrent fashion. And the fact that it gets clear down to around 1200 milliosmolar at the tip of the peritubular capillary that's running in parallel to the loop of Henle, helps prevent loss of the osmotic gradient in the interstitial fluid. You know, the fact that this is getting concentrated and is roughly in equilibrium with this helps preserve this concentration gradient. That is the main reason to think about countercurrent. It preserves the osmotic gradient. Now, the water that's coming out of the descending limb, the loop of Henle, can be picked up by the peritubular capillary as it goes back, and so the plasma ends up back in its dilute state. Just a quick question. Let me ask you to think about this for a second. What's going to happen to the red blood cells and the white blood cells as they get down to the bottom of the peritubular capillaries in the medulla before they get back up to towards the nephron, where the blood empties into the arcuate arteries? Are they going to swell and burst or shrink? The answer is they're going to shrink. This will draw water out of the red blood cells and they will shrink down, which they can stand. They can stand some shrinkage. The white blood cells too will shrink because of the extracellular osmolarity, but they will re-expand to normal by the time they get back up in the peritubular capillary. Okay, so this is kidney countercurrent. One of the hardest subjects to get your head around in the kidney that Morgan promised you was going to be hard. So far, we've learned about antidiuretic hormone affecting osmolarity, countercurrent preserving this osmotic gradient. The osmotic gradient is used by antidiuretic hormone to either concentrate the urine so it has high specific gravity, or dilute it so it has low specific gravity in response to how much we drink. Now, the question of how fast that happens is pretty intriguing to me. You know, I have to admit that if I drink a lot of water, my urine can get more dilute very quickly, within hours, or 5 or 6 hours. I could get fairly dilute urine. So I'm just guessing that the reabsorption of aquaporin can be pretty darn quick if it's needed. There must be fairly fast turnover of aquaporin in the apical membrane, but I still would expect it to be functioning on the minutes to hours time scale, not on the seconds time scale. Whereas deposition of aquaporin in the membrane to allow water out when you want to really concentrate your urine, that could happen in the minutes time scale. Okay, final question. It's going to be about aldosterone. I'll turn it off to make you answer the question blind. But you, of course, could cheat and look at the aldosterone slide in a third window, and I wouldn't be able to criticize you. Where and how does aldosterone stimulate sodium reabsorption? How is this reabsorption different from the constitutive reabsorption in the proximal convoluted tubule? Go. Last question. This end up taking 15 minutes after.
A little bit more, yeah.
Okay, last question. Maybe as I get better at this, I'll think of ways to highlight your good answers, because many of you have good answers, without drawing attention to those who don't know, because there's no shame in not knowing this yet. It takes time to learn and to get it. Constitutive reabsorption, we talked about in the first lecture that sodium and chloride 2 and water are reabsorbed, a large fraction of it, in the proximal convoluted tubule. Aldosterone fine-tunes the sodium reabsorption at the end of the path, at the end of the distal convoluted tubule. And the idea here is to, after you've got most of the sodium reabsorbed, let's just check and see if we have to fine-tune the sodium so that we get the right osmolarity. It's also important for governing the potassium concentration. And so it's best to think of it as a hormone that helps maintain homeostasis through a modulating effect. It's a cholesterol-based steroid, so it can get through membranes easily, get into the cells, bind to aldosterone receptors in the cytoplasm, affecting transcription of messenger RNA for proteins such as new channels and new pumps. The channels include the apical sodium channels that allow sodium to get in and be pumped through the sodium-potassium ATPase in the basolateral membrane back into the bloodstream. There are also potassium channels that allow potassium out. So if you start to get too much potassium, aldosterone will stimulate the production of more potassium pumps and more potassium channels to let potassium out. Remember back in the nerve section of the course, we talked about how hyperkalemia would cause membrane depolarization and eventually you get seizures. Well, that's the problem that we're preventing with this mechanism right here. Let the potassium out by getting more aldosterone. Aldosterone is released from the adrenal gland, the adrenal cortex, being a steroid hormone, under the stimulus of angiotensin 2, which is produced from angiotensin 1, which is produced from angiotensinogen, which is cleaved by renin. And that whole pathway we'll talk about next time. That's enough about it for today. It's called the renin or renin angiotensin aldosterone stress response. And it will, the final limb of it will be this hormone that enhances sodium reabsorption as a final touch to modify the urine composition and help maintain sodium homeostasis in the body. So today we've talked about sodium homeostasis and osmolarity homeostasis, and in that sense, water homeostasis. And we're going to continue down this path a little further next time, talking some more about regulation that the kidney provides. That's it for today. Any last questions? Thank you all for coming. Same drill on Wednesday, don't forget.
Thank you.
Thank you.